Good morning, everyone. It gives me great pleasure to welcome Dr. Cynthia Bulik, a long-standing colleague and friend, to UCSF's distinguished virtual visiting lecture series. Dr. Cynthia Bulik is a clinical psychologist and the founding director of the University of North Carolina Center of Excellence for Eating Disorders, distinguished professor of eating disorders at the University of North Carolina at Chapel Hill, and professor of nutrition and in the Gillings School of Global Public Health. She's also professor of medical epidemiology and biostatistics and director of the Center of Eating Disorders Innovation at the Karolinska Institutet in Stockholm, Sweden. Would have been there most of this year, but sadly COVID put the kibosh on that for the time being. Dr. Bulik received her BA from the University of Notre Dame and her MA and PhD from the University of California, Berkeley, just around the corner. She completed internships and postdoctoral fellowships at the Western Psychiatric Institute and Clinic in Pittsburgh. Dr. Bulik has developed eating disorders programs in New Zealand, the United States, and Sweden, and has active collaborations all over the world. She has published more than 600 papers and 50 chapters on eating disorders. She's the author of 7 books, including Crave, Why You Binge Eat and How to Stop, The Woman in the Mirror, Midlife Eating Disorders, Your Journey to Recovery, and Binge Control, a compact recovery guide. Dr. Bulik has been the recipient of numerous awards, including the Eating Disorders Coalition Research Award, the Academy for Eating Disorders Leadership Awards for Research and Advocacy, the Price Family National Eating Disorders Association Research Award, and the Don and Melissa Nielsen Lifetime Achievement Award from the National Eating Disorders Association. Dr. Bulik is the past president of the Academy for Eating Disorders, past vice president of the Eating Disorders Coalition, and past associate editor of the International Journal of Eating Disorders. She serves on advisory boards of several advocacy organizations and is the founder and co-chair of the Eating Disorders Working Group of the Psychiatric Genomics Consortium. Dr. Bulik's grand rounds presentation today is entitled Genes, Environment, and Eating Disorders, Accelerating the Science of Eating Disorders Through Global Collaboration. A very warm welcome to you, Cindy. And before we get going, just a reminder to everyone to please type your questions into chat, and Stephen Shaw will do the Q&A after the presentation. The floor is yours.
All right, thank you so much, Daniel. Thank you for the lovely introduction. You promised 1 minute, but it was actually 2.5. So hopefully I won't go over with my talk. Good morning, everyone. Thank you so much for being here this morning. I'm going to immediately share my screen and dive into the presentation. I need someone to give me a thumbs up that you are seeing my presentation and that it is advancing. All right, excellent. Okay, and one more thing. Okay, disclosures. First off, I'm a grant recipient and on the advisory board of Shire Pharmaceuticals, now Takeda, consultant for Idorsia, and an author for Pearson. And this is my gratitude quilt. So a lot of the research that you'll be hearing about today was funded by these various groups. And the talk map, this is where we'll go this morning. First, why study the genetics of eating disorders, understanding the current results, some guidelines for clinicians about how we can incorporate this into our practices, and a little bit about EDGI, the Eating Disorders Genetics Initiative. So just very briefly, the current topography of the feeding and eating disorders in DSM-5, all know anorexia nervosa, bulimia nervosa, binge eating disorder, and ARFID, the new kid on the block. We're going to be talking primarily about anorexia nervosa today, but also just a tad about bulimia and binge eating disorder. And what we know about them in terms of epidemiology is for anorexia, prevalence is about 1 to 4%, depending on the paper. Lifetime standardized mortality ratio, 5.4. So right now, aside from opiate abuse, it has the highest mortality rate of any psychiatric disorder. Bulimia, about 1 to 2% of the population. SMR, about 1.5, and binge eating disorder, 1 to 4% of the population, with a more even sex distribution, and also has elevated mortality. The heritability based on twin studies, Average is about 60% for anorexia, 60% for bulimia, slightly lower for binge eating disorder, around 45%. And presaging where we're going, one of the things that's really important about eating disorders is these are not mutually exclusive conditions. This is from the beautiful population-based data that we have in Sweden, that everybody who comes in for treatment the first time gets registered in a quality register, and then they get followed up annually. And this basically shows the large orange circles are their first diagnosis when they first register. And as you can see, although there's some stability in diagnosis over time, so people come in with anorexia and continue to have anorexia, you also see lines going all over the place. Luckily, some people remit, but also people change the nature or the presentation of their eating disorder, switching from anorexia to bulimia or from bulimia to binge eating disorder, for example. And this is one of the things that we're hoping genetics is going to help us unravel and be able to predict more effectively. So why study the genetics of eating disorders? And this is just a little bit about what fascinates me about anorexia nervosa. So first off, for people prone to anorexia, starvation is reinforcing. Most of us hate being hungry, and we don't like starvation for sure, but yet people with anorexia say that it's reinforcing. They say that at baseline, they're actually quite anxious, that starvation or restriction actually calms them, at least in the beginning of their illness. Fats are aversive. Most of us love fat, especially if you mix some sugar in with it, but people with anorexia say that the taste, the feel, the experience of fats are aversive. Activity is more reinforcing than food. Indeed, we have animal models, the activity-based anorexia model, in which we can get mice and rats to run themselves to death, even in the presence of their most favorite palatable food. And people with anorexia go through this perplexing hypermetabolic period during renourishment. We don't really know how to predict it, but based on any equation that we can have, their body burns more energy than one would expect. We don't know how long it goes on, and it takes a while for many people for their metabolisms to restabilize after renourishment. And finally, their bodies seem to revert to a negative settling point. And I use the term settling intentionally. You've heard about the set point associated with obesity research, saying that your body has somewhere that it likes to live. I don't really like the set point theory, but one of the things that we see is that once people with anorexia reach a really low body weight, even after renourishment, their bodies tend to pull them back down to their low weight. And we don't know biologically what's behind that. We tend to psychologize that process and view it as willful weight loss. I'm not convinced. So I've sort of termed this paradoxical response to negative energy balance. Negative energy balance being expending more energy than you're consuming, and energy expended is exercise, physical activity, rest, fidgeting, and purging. And people with anorexia prefer being in negative energy balance, while the rest of the world right now is tending to exist in positive energy balance, which is why we have a global obesity pandemic. The study of the genetics of eating disorders isn't just about the genetics of eating disorders. One of the things that we've already found and that we see across the board in psychiatric genetics is that knowledge about eating disorders genetics informs our understanding of many related phenotypes. Related phenotypes in terms of commonly comorbid illnesses like major depressive disorder, anxiety disorders, OCD, but also information about the genetics of obesity, of physical activity, of metabolism and nutrition. So the implications of our research go far beyond the understanding of eating disorders. So now, just for those of you who aren't geneticists, a super high-level tutorial on genome-wide association studies, or GWAS. Basically, we're passing over decades of work looking at candidate genes and linkage studies and going straight to the GWAS era, in which we get large samples of individuals with an illness or a trait and ancestrally matched large samples of individuals without the trait, or if it's a low prevalence disorder, unselected controls. And basically what we're doing is we're slathering millions of markers across the genome, and we're looking for areas on the genome or loci that differ between cases and controls. And when we identify those loci, it basically says this is an area where you need to look for potential risk alleles for the trait or disorder that you are studying. And the output of a genome-wide association study is called a Manhattan plot. And it's called a Manhattan plot because if you're lucky, the hits or the loci, those towers above the red horizontal line, are going to start looking like the Manhattan skyline. This is one of the famous now schizophrenia Manhattan plots. And when you read a Manhattan plot, you see the human chromosomes on the bottom, you see the significance level on the top. And since we're doing a million comparisons, we have to correct for multiple comparisons. So our significance level is 5 times 10 to the minus 8. So anything above that line, we consider to be a hit or a locus of interest. Now, the reason that it's really important to have a high significance level is once you decide that this is an interesting locus to investigate further, you're going to be spending a lot of money and you're going to be involving your neuroscientists and all sorts of other people to try to investigate sort of what the genes might be, what the pathways might be. So we want to make sure we're minimizing false positives. This is what's behind all of the psychiatric genetics research going on today, the Psychiatric Genomics Consortium. We have over 800 researchers and clinicians from around the world who are focusing on all of the major psychiatric disorders and understanding both genetic and environmental factors that influence risk. We are the Eating Disorders Working Group of the Psychiatric Genomics Consortium, which I founded in 2013, and we have been growing ever since. We have about 200 members now. We're meeting around the world or virtually whenever we can. And the biggest boost for the PGCED came with the Anorexia Nervosa Genetics Initiative, or ANGI. And this was funded philanthropically by the Klarman Family Foundation and included North Carolina, Sweden, Australia, with assistance from New Zealand, and Denmark. And the results that you're going to see next really are the fruit of the ANGI collaboration. So what we did, and it was sort of an amazing task, is ANGI collected over 13,000 samples from individuals with anorexia nervosa and 35,000 controls in about 3 years. And we then combined those samples with all of the rest of the existing samples and data that we had from around the world. But as you can see, ANGI makes up the largest component of our freeze-to analysis. And in total, we had 33 data sets with about 17,000 cases and 55,000 controls from 17 different countries. So analytically, it was complicated because we had so many different countries and different sizes of data sets that we combined. And this is our Manhattan plot, not exactly Manhattan yet because we only identified 8 loci, but those 8 loci in and of themselves are really transformational for the field because it's really the first time that we can say we have identified loci on the genome that are associated with anorexia nervosa. And as you can see in the different colors, the loci that we identified had been previously implicated in other genome-wide association studies. And they covered autoimmune disorders, metabolic traits, neuropsychiatric traits, and sex hormones. And this in and of itself is quite interesting because we see a lot of GI-related autoimmune diseases in some of these loci, IBD, ulcerative colitis, Crohn's, but also a lot of loci that are associated with anthropometric measures like BMI, like obesity, like body fat. So this was a very interesting first look at the loci, at the sample size that we had for our second freeze. But one thing to keep in mind is in these early stages of GWAS, when your sample sizes are still relatively small, you're going to see a lot of movement with these loci. So some of these ones that are just below the red line might start going above the red line as our sample size increases, but we might also see some of these movement in some of the loci that are closer to our significance level. So it's not time to write home about any of these yet. But the really interesting piece of this first GWAS that we published, not really the first GWAS we published, it was the first really interesting GWAS that we published, is that there's also valuable information below that red line. It's not just the loci that we're interested in. And a lot of the work that's been done on that information below the red line was done by one of my F1s, so Brendan Bulik Sullivan, my eldest, and he developed a approach called LD score regression that allows you to calculate genetic correlations. So not phenotypic correlations, but actual genetic correlations. And you can do this using LD score regression using published summary statistics. So you don't need to have the individual genetic data for every single person in your analysis. You also don't have to measure all of the traits on the same people. So when we did this back in the days with twin studies, we used to have a sample that included, for example, people who were evaluated for both eating disorders and depression, and then we could look at the coheritability or the extent to which they were co-inherited. But with LD score regression, basically what you're getting is the genetic analog of comorbidity. And I'll walk you through our genetic correlation results because they're fascinating. So this is first the overview, and then I'm going to break it down into bits. The way you read this is in the middle, you have 0. Everything to the right is a positive genetic correlation, which means the same genes are acting and their influence is in the same direction. And everything on the left side of the red line is a negative genetic correlation, meaning that the same genes are operative, but their function is going in the opposite directions. And now we're going to cut this down. These are only of hundreds of tests that we did. These are the ones that were significant after correction for multiple comparisons. So we're going to start out with psychiatric, educational, and physical activity. So these green, the dark green dots on the right side are the genetic correlations between anorexia nervosa and psychiatric disorders and traits. So we see the strongest genetic correlation between anorexia nervosa and obsessive compulsive disorder. And that comes as no surprise to people that work with eating disorders clinically, because we see that often clinically, that comorbid pattern. We also see positive genetic correlations with anorexia and major depression, schizophrenia, anxiety, depressive symptoms, and neuroticism. So this basically anchors anorexia nervosa genetically firmly within the psychiatric world and shows those strong positive genetic correlations with other psychiatric disorders. But then we also see positive genetic correlations with years of education and college completion. And I think this is really interesting. So educational attainment is the overarching trait. And this is interesting because for years, we have theories that go back forever about eating disorders families putting a lot of pressure on their kids and being very achievement oriented. But this actually shows that there's a positive genetic correlation between educational achievement and anorexia nervosa. So again, we're seeing a biological or a genetic explanation for something that we have very much psychologized in the past. And the same thing holds for the next finding. And this is a positive genetic correlation between anorexia nervosa and measured physical activity. So the study was done UK Biobank. It was measured physical activity with accelerometers, and we're seeing a positive genetic correlation with physical activity. So again, those of you that have worked with anorexia nervosa knows how hard it is to keep people with anorexia, or at least a subset of them, from moving. So even when they're severely emaciated, they're out exercising, or they're bouncing up and down in their seats in the unit, or just moving constantly. And again, we've often said that that's willful as an attempt to burn more calories. But what might actually be happening is there might be a genetic factor that is contributing both to the anorexia and to the increased physical activity. Now, the next pattern, all on the negative side with one exception, starts showing genetic correlations between anorexia and various metabolic factors. So we're seeing negative genetic correlations with insulin resistance, fasting insulin, leptin, and type 2 diabetes. And the only positive genetic correlation with metabolic factors is with HDL cholesterol, or a favorable metabolic parameter. So not only does anorexia have these psychiatric genetic underpinnings, but there also appear to be metabolic factors, and we're not seeing these in other psychiatric disorders. And then finally, we see another set of negative genetic correlations with anthropometric measures. Body fat percentage, BMI, hip circumference, waist to hip ratio. So negative genetic correlation between anorexia and all of these anthropometric measures. Now, this made me rewrite my conceptualization of anorexia because people have always asked me, is anorexia the opposite of obesity? And I always said, no, anorexia is psychiatric, BMI, obesity is sort of metabolic and behavioral. But according to these genetic correlations, we have to look at the fact that there may also be an anthropometric component. We might actually be looking at, to some degree, opposite ends of the BMI spectrum from a genetic perspective. And then if you take this whole panel together, this sort of gives you the genetic topography of anorexia nervosa. And it has led us really to sort of reconceptualize how we think about anorexia nervosa etiology. And what we've done is we've actually coined the term Metabo-Psychiatric illness to account for both the anthropometric, metabolic, and psychiatric genetic components. And it helps explain this perplexing ability to reach and maintain a low BMI in a world where most people struggle to lose a few pounds and most people's sizes are increasing. It may explain this frequent return to a negative settling point. So the number of times that someone comes into a hospital, is renourished, gains their weight, and then as soon as they're discharged, their body pulls them back down again. And this could go back and forth, leading to a revolving door pattern. Perhaps some of these genetic factors can explain what that metabolic factor is. We have these negative genetic correlations with BMI and other unfavorable metabolic parameters, and the positive genetic correlations with HDL. And it might also help us explain this paradoxical reaction to negative energy balance. Why is it that someone who is genetically prone to anorexia nervosa and doesn't have the illness yet, but the first time they go on a diet or they lose weight secondary to an illness, that becomes reinforcing to them? Whereas for other people, it's a horrible experience. Diets tend to be really miserable for most people, but people prone to anorexia find calmness and a reduction in anxiety when they reduce their caloric intake. And where we go from this are, maybe we need to reconceptualize anorexia as a metabo-psychiatric disorder, and greater attention to the metabolic factors may improve outcome. We are not great at treating this illness. When we look at the data, still only about 30% of people with anorexia nervosa achieve full recovery. I've already mentioned the high mortality rate, and a large portion of people sort of have this middling result where they're not really well, but they're not really ill, and their BMI sort of hovers around low weight, normal weight, and they still have a lot of the anorexia cognitions. We absolutely have to improve that, and maybe looking at metabolism more will help us understand the underlying biology. It could explain why adequate refeeding is essential to preventing relapse. In this country, we have this horrible situation where people are often punted out of hospital early, sometimes when they reach 80–85% of their ideal body weight, and that is a recipe for weight relapse or losing weight again when they're discharged. Other countries have the luxury of keeping people in until they reach BMIs of 20, sometimes 21, and often their experience is not the same as ours of going back and forth between low weight and refeeding, low weight and refeeding. And finally, ultimately, we need to understand the metabolic mechanisms. It's cool that we can see this panel of genetic correlations, but we don't know what they are yet. And increasing sample size and clarifying some of the pathways associated with the metabolic differences are going to be essential to understand this aspect of the illness. And then taking it one step further, up till now, I've only talked about anorexia nervosa because those are the only GWASs that we have. We do not have a GWAS of bulimia and binge eating disorder yet. But there are some other tools that we can use to see if the DSM feeding and eating disorders chapter actually carves nature at its genetic joints. And I'm going to talk a little bit about polygenic risk scores now. And again, we're not just looking at those loci, we're really looking at all those other bits of information below that significance level that still contribute to risk or to the phenotype of interest. And basically a polygenic risk score, you need two samples to calculate polygenic risk score, a discovery sample on which you create the polygenic risk score, and then a target sample in which you use the polygenic risk score. And basically what it is each individual gets a single score, which is a weighted sum of all of their risk alleles. And then you can basically determine how high someone's polygenic risk for an illness is. You know, are you in the 90th percentile, in the 10th percentile? Where do you rest in this normal distribution of risk based on your risk alleles, based on the summary statistics from a genome-wide association study? And it's highly useful. And I put this paper down here because I think it's one of the most eloquent papers out there that describes the potential utility of PRS now and in the future. And first, can we actually improve risk assessment by combining polygenic risk scores with other measures of risk? Second, can we use PRS to screen for mental health disorders in the population? Is this a direction that psychiatric genetics is going to go in? Third, can PRS help in making a diagnosis or making clinical decisions? And if I go back to that early slide with the bubbles, you know, when we think about when that person walks into our office for the first time, let's say at age 13 with anorexia nervosa, we do not know. We cannot predict if they're going to develop a severe and enduring course of the illness, if they're going to cross over to bulimia nervosa at some point in time, or if they're going to sort of have a more of a drive-through illness where they might be ill for a couple of years and then they recover. PRS might be able to help us sort of get early warning signs of what the potential clinical outcome might be, and then we can tailor our treatments accordingly. And are psychiatric polygenic risk scores associated with treatment response? And ultimately, especially when it comes to medications, this might be really important. You know, right now for anorexia, we have no FDA-approved medications for this illness. And it goes without saying that we have no medications that actually target the underlying biology of the illness. But hopefully, as our research progresses, we'll get to the point where our toolbox becomes a little bit more full, and we might find that certain PRS patterns predict response to one drug better than another, or even to one psychotherapy better than another. And then are PRS associated with adverse physical health outcomes in mental illness? For example, can we use polygenic risk scores to predict those individuals who are more likely to have really dangerous weight gain associated with antipsychotic medication? And these sorts of questions could also be put toward the treatment of anorexia and bulimia and binge eating disorder. So a great paper, I highly recommend it. Give it a read. So since we don't have a GWAS of bulimia or binge eating disorder, we turn to the UK Biobank, which is an amazing global resource where for 17,000 people, we have genotypes and tons of anthropometric measures as well as psychiatric diagnoses. And we identified 768 people with anorexia, 423 people with bulimia, and 561 people with binge eating disorder. Now, we already know that these sample sizes are way too small for a GWAS, so this is just a polygenic risk score results that we're going to see in the next couple slides. So again, and I'll break it down piece by piece, but what you're going to be seeing is genetic differences across anorexia, bulimia, and binge eating disorder. So really the first look at how these three illnesses differ on a genetic level. Next several slides, anorexia is sort of like this lightish blue color, binge eating disorder is purple, and bulimia nervosa is magenta. So we're going to start with psychiatric traits. And I'll walk you through this. Any of the dots that are filled are significant. Again, one is the no, these are odds ratios. So one is no effect. Everything to the right is increased, elevated odds ratios. Everything to the left is protective odds ratios. And I'll walk you through one by one. So the first thing we did, this is sort of a sanity check. We took the anorexia PRS that we derived from the study that you just saw and applied it to the UK Biobank samples. And as you can see, the only significant odds ratio was anorexia was associated with anorexia. So the anorexia PRS did not predict bulimia and it did not predict binge eating disorder. So that's the first sign, A, that our anorexia PRS is working, and B, that there is a difference across these three disorders. But the differences continued. So for schizophrenia, we see a positive association with binge eating disorder and anorexia, but not bulimia. For major depressive disorder, we see a positive association for binge eating disorder and anorexia. For autism spectrum disorder, even though these aren't significant, I highlighted them because they're in different directions. So we see a positive association with binge eating disorder, bulimia, and a negative association with anorexia nervosa, which is interesting because there is a whole literature on autism spectrum and Asperger's being associated with anorexia after recovery, but that's not looking as solid as it might have in the past. Whoops, I think I missed one. Yep, and then the final one is ADHD significantly associated with binge eating disorder, but not the two other disorders. And that is consistent with what we're seeing epidemiologically, where we see more attention deficit hyperactivity disorder in people with binge eating disorder than in people with the other eating disorders. And then the next slide is looking at the metabolic factors. And I'm only going to point out one thing here because these aren't significant, but the pattern is interesting. And this is the pattern associated with binge eating disorder. So you see increased risk for type 2 diabetes, low HDL cholesterol, and then high fasting insulin. So the pattern that we're seeing with binge eating disorder very much mirrors what we see with obesity. And we don't see that same pattern in anorexia nervosa and bulimia nervosa. So right away, you see a marked difference across these three different disorders on the metabolic side. And then finally, and I think this one really jumps out at you, it's the anthropometrics. And we're looking at polygenic risk scores for waist circumference, BMI, et cetera. And as you can see, very much reflecting what we saw in the previous study, all of the anorexia nervosa, almost all of the anorexia nervosa odds ratios are negative, whereas all of the bulimia and binge eating disorder odd ratios are positive. So I think what we might be seeing is underlying eating disorders. There is a shared psychiatric component, but there are differences when it comes to genetic factors that influence metabolism and anthropometric measurements. So this is really the first glimpse of how these three disorders might differ. Power was low. We absolutely need GWAS for bulimia and binge eating disorder. We're getting there, but this is a pretty solid and interesting first glimpse at what might be going on. So to what end? We might be able to identify subtypes of illness based on PRS. One of my predictions is that when we get larger sample sizes, we're actually going to find that there are some anorexia nervosa cases that are more psychiatric, and there are some anorexia nervosa cases that are more metabolic. And ultimately, that might help us predict what type of interventions we might need to use to be more effective. It might predict likely course of illness and tailor treatment accordingly. And it could move us toward a more personalized medicine approach to treatment instead of a one-size-fits-all. And ultimately, we hope to be able to identify biological pathways so that we can move forward with drug repositioning or development based on our genetic results. And finally, and this is sort of one of my passions because it still happens far too often, is we have to eliminate mortality from anorexia nervosa because far too many people still die of this illness, and it is embarrassing that it still happens. So how do we take this information today and use it in our practices? And this is really important. Just yesterday, I got an email from someone out in the Bay Area, actually, who said, you know, my daughter has a double mutation in this gene, and I know it causes eating disorders. And, you know, those sorts of emails just make my head explode. But yet, they tell me exactly what our responsibility is, because there are lots of companies out there that are saying things like that. And we need to make sure that that misinformation doesn't get into the hands of patients. So this gets into sort of science communication about genetics. And I think we all know that anti-intellectualism and anti-science are rampant right now. And the Clorox and coronavirus is one of those examples. I consider it to be the scientist's responsibility to interpret what we do and to contextualize what we do. And it's the clinician's responsibility to have a general understanding and to assist patients and families. I know our patients and families are educated. They bring research to us and they want us to know what's going on and help us to understand how it applies to them and their own personal narrative about their illness or their child's illness. There's a very important role for genetic counseling, and we underutilize genetic counselors in this country. And some of the messaging do's and don'ts, and this is leading up to sort of a simplistic but effective way to talk about this with patients. First off, genetics is just one piece of the risk puzzle. If anything, doing genetic research is going to help us understand the environment more. We have to get completely away from all or nothing thinking. Whenever someone says genes or environment or nature or nurture, I cross out the or and I put it in and, because we know it's both and we need to think about both when we develop illness narratives. People are challenged to understand probabilities. We like things to be all or nothing. It's much easier to think that way, but yet we have to get people thinking in terms of probabilities. We have to move people away from genetic destiny. That's a trap. We don't want people to think that we hear so often just because a mom or a dad had anorexia that their kid is destined. And another one that we have to be careful about is genetic guilt. We start hearing people feel guilty about the chromosomes that they passed on, the genes that they passed on. And if there's one thing we have no control over, it's what gets passed down at the moment of conception. And also just the misinterpretation that eating disorders are genetics and not psychological, with the caveat there being the eating disorders are genetic and not psychological, so I don't have to work in psychotherapy or I don't have to do FBT. Not true. Those are the only tools we have right now. We do not have a way of modifying genetics to improve illness. And then genetic simplification, like the one that I just mentioned from the email last night. I have the genes for eating disorder. Or if you ever see a headline in a newspaper that says, gene for anorexia discovered, it's not going to happen. These are complex traits. There are going to be hundreds of genes and hundreds of environmental factors that influence risk. And genetic testing. We are not doing this research to lead toward a genetic test. And polygenic risk scores run the risk of being misinterpreted as genetic tests, but they're not. They're just relative measures relative to the rest of the world. What is your polygenic risk for this illness? And they also change as the research gets more mature. So as our sample size gets larger, polygenic risk scores become more robust and they're going to do better at predicting risk. So this is just a model that I use sometimes to talk about genetics with patients. And I basically say that, at conception, we get shuffled. We get all of the genes from the sperm and all of the genes from the egg, and they get shuffled around. And basically, our ultimate risk comes down to 4 buckets. The spades are genetic risk factors, but they're also protective genetic factors. So even if you have handed down a lot of genes that are associated with anorexia risk, you probably also have some genetic protective factors, and the partner, whoever presented the other side of the 50% of the genes, probably gave you some protective factors as well. But then those get combined with environmental risk factors, bullying, teasing, being in a sport that emphasizes thinness. But then there are also environmental protective factors. Like if you have an awesome ballet teacher or coach that focuses on strength instead of on thinness or thin to win. And it really is the combination of these things that lead to an individual's risk. Right now, we can't do anything about the spades and the clubs. Those are fixed. They're fixed parameters. The only thing we can do things about are the hearts and the diamonds. And the only thing to add to this is, for some illnesses, we also have these sort of rare variants or CNVs that if you have them, you develop an illness. But up to this point, we've identified no rare variants and no CNVs that are associated with anorexia nervosa. So in general, on a very high level, and of course it'll break down if you dig too deep into using this model, but it is a nice way to sort of explain the 4 areas of risk. And so genetic counselors will use examples like this to talk about the risk to offspring. And genetic counseling for mental illness has been shown to help alleviate stigma and shame, correct misconceptions about conditions, prepare family members to intervene, promote help-seeking behaviors. But there's only been one study on genetic counseling and eating disorders, and that's a study we did with Julianne Michael at UNC Greensboro, where she studied 170 individuals with a personal history of an eating disorder. And one of the questions we asked is, what do they think? Since they had an eating disorder, what do they think the risk to their children was? And this was the estimated perceptions of probabilities of recurrence for children of individuals with eating disorders. They assumed that their kids had a 57.6% risk of developing anorexia if they were a girl, a 43% risk if they had a boy, but the empirical data suggests that it's really about 7.5%. And 100% of people assessed overestimated their risk to daughters. So this again it just underscores in a really simple way how important science communication is, because people believe that once they have an illness like this, their kids are going to get it. And it's not that simple. And this is just, we're not going to go through the detail. We're just going to look at yellow versus blue all the way down. And what we did is we gave these 107 people an imagined experience with a genetic counselor, sort of talking about all the things we talked about this morning, about what influences risk, about genetic risk, genetic protective, environmental risk, environmental protective, and then we did a pre and post test. And just getting that information led to reductions in stigma, in shame, in looking down upon the person with the illness, and guilt, and in like treating that person differently for being different. So there was a definite impact on just being walked through the accuracies and the truth about genetic transmission to offspring. And just one example, person said, wow, just reading this validated so many things I felt, like it is genes and environment, and that guilt is so common in people with eating disorders. I feel like people need to hear and understand this, people with eating disorders and the public in general. So it's our responsibility not just to do the science, but to package the information for patients and families. Clinicians should make an effort to integrate genetics into their own case conceptualizations and develop comfort with answering their patients' questions without expecting themselves to become experts in genetics. And know your limits. And if you live in a country with genetic counseling, which we do, make use of your colleagues. And if not, find resources for your patients. And don't perpetuate misinformation. We need to start education at the very basics, especially in med school, to make sure that people are not being taught old theories that no longer hold water. So we really need to update misinformation for the public as well as for healthcare professionals. And then finally, my last bit is EDGI, or the Eating Disorders Genetics Initiative. And this is a study where we're going beyond anorexia nervosa, which we did at Angie, went from Angie to EDGI. We have added bulimia nervosa and binge eating disorder, and then ARFID is pending. We really wanted to add it, but NIH didn't feel like the phenotype was ripe enough yet for investing a lot of money in a genome-wide association study. And what's different about EDGI, and what's slightly insane about EDGI, is our global goal is to sample 100,000 individuals with eating disorders from around the world. And NIMH is supporting the United States, Australia, New Zealand, and Denmark. The United Kingdom has found independent funding. I've gotten independent funding in Sweden. And we're bringing Mexico, the Netherlands, Puerto Rico, Taiwan, Italy, and more countries on board. So this is a truly global effort. And embedded in here is the importance of trans ancestry GWAS. So much of the psychiatric genetics that has been done to today and for many disorders comes from European ancestry populations. And our goal, obviously, is to make sure that we're broadening that to really capture eating disorders around the world, because we don't know if the same genetic factors contribute to eating disorders similarly across different ancestries. For EDGI, we're asking the same questionnaires around the world, so it's harmonized phenotyping. We're diversifying the samples. Everything is digital, so digital consent, digital questionnaires, a simple at-home saliva collection that's totally COVID safe. We're engaging the advocacy community, and we're engaging clinicians. So we're really trying to create a research community so that everyone around the country, around the world, is encouraging people to participate in EDGI. The domains that we're assessing: life events, trauma, physical activity, OCD, alcohol and drug use, tobacco use, anxiety, and depression. Because, you know, one of the things that we can do is really can start understanding, once our sample size gets big enough, you know, why some people who are genetically predisposed to an eating disorder don't develop one. What are the protective factors? Or we might also find people who have really low polygenic risk who do develop the disorder, and then we'll be able to see what life events might trip someone, might be the tipping point for someone who actually has low genetic risk to develop an illness. This is just a list of the different diagnostic instruments that we're using. Again, all online, so people can just sit down and do it all at home. This is our website. All you have to do is take the survey to be screened, and if you pass the screening, then you can go on to do the rest of the questionnaires, either in English or in Espanol. So we've got everything in Spanish as well. We have launched around the world. So we launched in New Zealand, we launched in Australia, we launched in the UK, and the next launches that we're going to have are Puerto Rico and Mexico. So, you know, these are big events where we do, you know, large scale social media and media outreach to engage people in the effort. And I've developed a talk show, a Facebook talk show, where we're bringing people in, all sorts of people who have experience with eating disorders and body image problems. This is more of an educational talk show, but it's also promoting EDGI and encouraging people to participate. And this is the team. So the EDGI team, UNC is the center, and the folks on top are all of my colleagues in the PGCED. Without them, none of this research would be possible, and it's impossible to name them all. But it's an amazing group, and you all are invited to join if you have eating disorder samples. And follow us and help recruit for EDGI. We're on all social medias at EDGI Study, or in Spanish at EDGI Estudio. And follow us on Twitter as well. We try to get lots of good information out there and really practice what we preach, and take this research and package it and disseminate it well, not just for scientists, but also for the public and for the patients and families with eating disorders. And someone just wrote on my PowerPoint, which is interesting. And that's all for me today. Thank you so much for your attention.
Thank you, Cindy. That was an amazing talk. You more than made up for the minute over on the intro that Daniel went. So we're in good time, and we got some interesting questions. So the first one that came in was back to your initial slides. Really an interpretive question. What do you mean to be a negative correlation between anorexia and metabolic factors? And those are negative genetic correlations. So the higher your genetic risk for an eating disorder, your genetic risk for metabolic factors would be on the opposite way. Do you just want to clarify that, if I say that quite right?
Yeah, sure. So it basically means they are genetically correlated, which means that the same genes are operative. But for anorexia, they increase your risk. But for example, for type 2 diabetes, they decrease your risk. So the effect is in the opposite direction.
Except for HDL was
except for HDL, exactly. And that's really interesting, because that's the only favorable metabolic parameter that came out of all of the metabolic parameters that we looked at.
I will say before I go to the next question that many years ago, when I had left my postdoc at UCSF and was a visiting assistant professor at UC Berkeley, there was this very bright grad student with kind of red hair named Cindy Bulik, who took a course or two of mine, and we chatted about career, et cetera. And I was out to North Carolina about 12 years ago and gave a talk, and we met up. And I wish you could be out here in person rather than this kind of a Zoom connection, but thanks for joining us. So next question from Aaron Accurso. What is your hypothesis about correlations with these anthropometric or metabolic components in atypical anorexia nervosa? And you might want to define for those who don't know what atypical anorexia nervosa is.
Yeah, that's a super great question. So atypical anorexia nervosa is complicated. It is currently defined as meeting all of the criteria for anorexia nervosa except the low weight. And I'll go into the complicating factor first. So this could mean that someone has a BMI of, say, like 21, and they lost a lot of weight and they got down to a BMI of 21. It could also mean that someone has a BMI of 40 and lost a lot of weight, and they're still in the overweight or obese category, but they have all the characteristics of anorexia nervosa. And NIMH wouldn't let us include atypical anorexia in this because the definition is still a little bit unclear. But this is the nice thing about having other countries involved. So both the UK and Sweden are including atypical anorexia nervosa. So we're going to be able to answer that question empirically. I think what we're going to find, this is just my hypothesis, is we'll see the exact same genetic correlations with psychiatric disorders, but the metabolic ones will look more like bulimia and binge eating disorder. I think it's that metabolic piece that's going to distinguish atypical from typical anorexia nervosa. And it will also help us decide whether this is a good name for it or not. In my own mind, we just finished a Delphi study that is asking people what the next steps in understanding atypical anorexia nervosa should be. And there's so many mixed feelings, and I'm using the word feelings on purpose in the field about this label, because on one hand, people who have what's now called atypical anorexia nervosa are ill. And they deserve treatment, and they might be starving, even though they're in the obese range. But calling it anorexia nervosa might not be accurate. We might need to develop a separate name for it. And if we find that there are real genetic differences, that might actually compel us to find a different name for it. But it deserves and should be legitimized as a condition. That's sort of my bottom line on the clinical side.
All right, thanks. Great answer. And from Titileo, this is a moderator kind of stratification question. Any findings on race ethnicity correlates or disparities with in relation to the data you presented?
Right. Nope. The data that you saw were 100% European ancestry. And that is precisely what EDGI is designed to do. We just submitted our first report, and we're really excited. We hit our minority targets, as NIMH calls them, for all three of our current sites, US, New Zealand, Australia. We have to change this. In some ways, it made sense for the PGC to start with European ancestry populations, but now we have the ability to do cross-ancestry GWAS. We know how to deal with those data, so we have absolutely no excuse. And we have chips, we can genotype better now that capture different ancestries. So there really is no excuse at this point not to have diverse samples. And part of that is, it's very similar to what we're seeing with vaccines and encouraging people to take vaccines. We need to develop trust in ourselves as researchers so that underrepresented people feel comfortable participating in this research. And we're doing a lot of work in figuring out how best to get this information disseminated and to get people comfortable with participating globally.
Thanks. So from Sarah Forsberg, whom I now see on the screen, but I'll read it quickly. We often see ongoing GI issues well into recovery, despite often communicating that many of these issues should resolve over the course of renourishment. How might genetic findings inform what we tell patients and our treatment of GI issues? Any insight, for example, from your microbiome research?
Yeah, not even necessarily microbiome, and that's another talk. But if you remember our Manhattan plot, a couple of the loci were associated with autoimmune GI disorders. And I think clinically, often people tell folks that you're going to have GI issues after your eating disorder, and it's the eating disorder that might have caused them. And although that might be true, we're also seeing these genetic correlations as well as loci that also contribute to GI illnesses. And this is actually a flourishing area of research because on a population level in Sweden, we're also seeing a lot of GI illnesses before the eating disorder takes hold. So there's a bi-directional relationship between some of these autoimmune GI disorders and eating disorders that we really need to do some more biological unpacking with, and they don't necessarily resolve. And it might not be because of the eating disorder, it might be because these people are biologically prone to both eating disorders and GI problems.
Yeah, fascinating. Let's see, are there any genetic correlations between eating disorders and food preferences or food taste in the general population?
Great question. We don't know. We have a couple studies that we're doing in Sweden right now that are more on the fMRI side of my work that are actually looking at those things. And that would be especially interesting when it comes to the fat preference piece that I talked about really early. What's behind this just aversiveness toward fats that is just so uncharacteristic of most humans and animals. But no, I do not have any more information on that than what I presented. Good idea.
Let's see, at what point do you think NIH might fund studying the ARFID genome? And are there certain papers that need to be published before they feel this diagnosis is well enough established?
Thank you for asking that question, and please publish the papers. Yeah, so we just had a talk with them last week, and they want to see, their biggest concern is that right now clinically, ARFID has these 3 different subtypes. It's the tactile, the aversive, and the non-appetite. I missed one. No, the bad experience, so more phobic avoidance. And they're concerned that if we sampled ARFID now, we would have an inadequate sample size to cover all 3 of those. Now, whether those 3 actually exist clinically is not entirely clear. A lot of people who work with ARFID say that they sort of see elements of all of those across their different patients. It's not like they clinically assort into these neat little groups. So whatever data we can have clarifying clinically and epidemiologically what ARFID looks like, and of course, a twin study on ARFID would be wonderful. That's the kind of sort of foundational work that we need in order to convince them that it's time to move forward.
So for those not in the know, could you define the acronym ARFID?
Avoidant restrictive food intake disorder.
Right. So I've got a question for you, Professor Bulik. So
it's always scary when you're old supervisors, and I have a question for you. Yeah.
So how do you think about in the genetics consortium and in eating disorders, research-wise and clinically, what the RDoC brings? I mean, so you've got categorizations of AN and BN and binge eating disorder. And the thought is that looking at underlying dimensions and looking at neural circuits and all the way up levels of analysis to psychosocial and cultural factors will help us more than these categories. How do you think about that?
I thought about that deeply when we designed EDGI. If you remember the picture with the tree in the middle and the different domains, what we tried to do is really, we thought about RDoC when we thought about RDoC and we thought about comorbidity when we designed our assessment battery. And we wanted to make sure that if we get this harmonized assessment battery across the world in, 100,000 people, we'll be able to get away from just doing these disorder-focused GWASs, and we'll be able to start looking at those underlying dimensions and the genetics of the underlying dimensions. And the best example is the next freeze that's going to come out of the PGCED is going to step back from diagnoses and just look at a behavior. We're just going to look at it. We're going to do a GWAS of binge eating. And that cross-cuts all 3 of the illnesses because you see binge eating in anorexia, in bulimia, and in binge eating disorder. And then, that's down to the behavioral level. And then, of course, there's another level above that where we can go, even more abstract, for lack of a better term. But I think there's going to be a lot more information and a lot more statistical power when we get away from just doing disordered focused GWASs.
So we're at our time. I want to say that we are unintentionally, Cindy, the both of us, plagiarizing each other in terms of genes or environments related to things like eating disorders or ADHD or oppositional defiant disorder. I think the human mind is kind of wired to think, well, it's biological or it's psychosocial, and it's a both and, not either or. And if we could erode that thinking and finding ways to make, just like with coronary artery disease and substance abuse, there are genes that make you vulnerable, there's experiences and there's choices and there's settings and cultures and contexts. If we could get the population to think more, and I think we do a lot to reduce the stigma and get treatments more out there for everybody. So thanks for a wonderful talk and great response to questions. A round of virtual applause for Dr. Cindy Bulik. Thanks for being our distinguished visiting lecturer.
Thanks for having me.
All right.